Students, I need to teach you a little bit about a few of the organelles, and we're going to do it through a video. First concept: structure leads to function. So today, you should be able to see how this is true. There's a couple of specific structures. We're not going to review all the organelles, but we're going to review a couple that have some very interesting structures. And those structures should make sense to you as to why they're structured that way. So let's begin with the mitochondria. Okay, first structure is this outer membrane. A couple other comments. Notice the shape and size of this is similar to a prokaryote, and that is one of the lines of evidence that this used to be a prokaryote. We've got an outer membrane and an inner membrane. And this, again, is another line of evidence. Recall that when the prokaryote was engulfed by the eukaryote, it gave the prokaryote another membrane, right? And that's, if you picture it folding in, you can kind of use your imagination. I would refer you to our notes from last week for that. But anyway, when this prokaryote was engulfed by the larger cell, it acquired a second membrane. So we got both an inner and an outer. The inner membrane has some specific features, and we're going to zoom in on that to see just what's embedded on that membrane. So let's do like a little microscopic kind of zoom in thing, where you're like zooming in on that particular inner membrane, and we can see the inner membrane here, okay? And then we have the outer membrane here. And there's this space in between the inner and outer membrane, this little gap. And it's filled with a high concentration of protons. This is the key function of the mitochondria. The number one function is to produce ATP. And there's this little ATP synthase protein, ATP synthase, that's embedded on the inner membrane, right? This is the inner and this is the outer. So know the location of the ATP synthase, know that it's located on that inner membrane, and that when the protons flow through, ATP is produced. In other words, the flow of the protons from a high concentration to a low concentration provides the energy to phosphorylate the ADP and basically take the phosphate, add it to the ADP, and then you produce the ATP. Normally, that reaction always requires energy, and so again, this flow of protons is what provides the energy to catalyze that reaction. They flow through, kind of like I like to think of like a hydroelectric dam. When you got water flowing through, you can harness the energy from it. Just like we've got over in Folsom, right? We got the water behind the dam. You let the water flow through, you can harness the energy. It's just analogy. It's not a perfect analogy, but hopefully that helps a little bit. Okay, so that's one of the features there. There's also this space in the middle of the organelle called the matrix, and it's like the fluid, okay? But there are some reactions that take place in the matrix. For example, the Krebs cycle occurs here. You don't need to know the details of the Krebs cycle yet. You will in Unit 3. Lastly, we've got these cristae, folding of the cristae greatly increases the surface area, which leads to more efficient ATP synthase. So just think of it this way, right? You got a limited space to put these. I'll refer you back to here. You've got a limited space for where you can place these ATP synthase proteins. But if you've got a lot of folding, then you've got more room to put those ATP synthase, right? There's just more area for these reactions to take place. And so this is the mitochondria's way of just being more space efficient and getting a more efficient ATP production. This folding really dramatically increases the surface area. Remember, that's kind of a theme of this unit. We've talked about surface area before when it comes to cell size and stuff. So, okay, so that's enough about the mitochondria. Let's go ahead and take a look at the chloroplast in more detail. Maybe pay attention to some of its features. The chloroplast is similar to the mitochondria in that it too is believed to have been a prokaryote. So it also has this double membrane. Remember, the theory is called endosymbiosis, right? So you got a double membrane. Now, another feature of the chloroplast is that you have this stack of structures. The whole stack together is called a grana. And then the individual parts that compose the grana are thylakoids. Now, the thylakoid happens to be kind of, well, let's say a few things about it. It is the location of the light-dependent reactions, and it contains chlorophyll. It too has a very high surface area, right? If you look at the structure here, you got all these folds, right? All these folded membranes, and that has the effect of increasing the surface area. And of course, that's just another way of increasing the efficiency of these reactions, right? You got more surface area, you got more room to put the proteins and catalyze the reactions. This will make more sense when we zoom in on the thylakoid, which we're going to do right now, actually. So go ahead and take, like we did before, we're going to go ahead and zoom in on an individual thylakoid, and we're going to draw it, what that would look like if we zoomed in. So you can do your little thing again if you want. So if we zoom in on that thylakoid, what we would see, right, is we'd see this, this is like, I guess you'd call the lumen of the thylakoid, which is the inside space. So the lumen would be like in here, right? But then it's encapsulated by two membranes, right? Got a membrane here and a membrane here. So the lumen of the thylakoid, similar to the cristae of the mitochondria, it too has a big accumulation of protons. The accumulation of protons will provide us the power to produce ATP and other energy carriers. More details on that to come in a later unit. But right now, just know that the lumen of the thylakoid is where we build up the protons. In the membrane of the thylakoid are these chlorophyll pigments. So label those as And if you recall, chlorophyll pigments are what capture the light energy. So if you want, you can have the light energy coming in, being captured by the chlorophyll pigments. And then out of this reaction comes oxygen. If you recall, photosynthesis brings in sugars. I'm sorry, brings in CO2 and produces oxygen and sugars, specifically glucose, C6H12O6. Okay, you will get all the details on that later, like I said. But right now, the focus is the structure and the function. So again, just to recap, the folding, this provides a lot of surface area for pigments and a lot of surface area for the proteins that catalyze these reactions. Okay, so he says this is where the light-dependent reactions take place, which is where you've got light being converted to chemical energy. Okay, well, what about in here, this space, the stroma? What's going on over here? So as we said, we've got the light-dependent reactions taking place in the thylakoid, which means that we have the light-independent reactions taking place in the stroma. Reactions. Specifically, the Calvin cycle. And this is where the carbon fixation takes place, right? CO2 molecules are reacted to make C6H12O6. All right. I think that's about all we need to say about the chloroplast so far. Carbon fixation occurs in the stroma. The capture of light occurs in the thylakoid. Okay. Alrighty. Next, organelle. One more organelle I want to talk about. Has an important function. That is the lysosome. Maybe in middle school you were told that this is the stomach of the cell. It's true. It's a good analogy. The reason we would call it the stomach of the cell is because it's filled with acids and hydrolytic enzymes. Hydrolytic enzymes. This means that those enzymes are going to catalyze hydrolysis reactions, which is basically the splitting up of larger molecules. You're going to break those larger molecules down into their monomers. So we can say that if we want, catalyze hydrolysis reactions. So that would look like this, right? You got like a polymer, and by the time that the reaction is taking place, you will have split it into its monomers. So this is breaking down food and waste. So this would include all of those biological molecules, breaking down proteins into amino acids, breaking down carbohydrates into their monosaccharides, breaking down nucleic acids into nucleotides. Those are just some examples of hydrolytic reactions. So the inside is very acidic, which means that you've got a low pH, right? And the enzymes only function at those low pHs. Right, and this is actually a really important idea that I want to get across, and that is this compartmentalization. This idea of compartmentalization that is unique to eukaryotes allows for extreme pH in an isolated part of the cell. So, in other words, a prokaryote would not be able to produce extreme pHs because it would damage itself, right? Like if you produced a really acidic environment inside the prokaryote, the whole rest of the cell would be affected by that because there's no way to isolate the acidity. So the acidity is isolated in the lysosome. So the rest of the cell is protected, right? So we can get extreme conditions in these little compartments without messing up and affecting the rest of the cell. Okay, what would happen, right? What would happen if a lysosome split open and poured acid into the rest of the cell? Well, it would denature everything. Like it would denature the proteins. It might denature the genetic material as well. So we've got to keep it encased inside the lysosome. But by the way, the lysosome is structurally, it's just a small spherical organelle. Very simple structure, right? Okay. That's going to wrap it up for organelles.